It was 6 years ago when Dolly, the first mammal to be cloned from an adult cell, passed away. This achievement, it was a great milestone. It was considered the first of the first and paved the way for the future. But what if it had been a human that was cloned? That would cause a huge controversy, man. In this What's Up Cloning 101 video, I'll be explaining to you how cloning works, the types of cloning, the advantages cloning can bring to us, and the ethical issues it is facing. First things first, what is cloning? Cloning is a complex process that produces an identical copy of genes, cells, or animals which has the exact genetic or inherited traits of the original, also called the donor. Species that scientists have successfully cloned include cattle, swine, sheep, mice, rats, rabbits, cats, mules, horses, and one dog. That's probably more than you imagined. Now, you'd think cloning is something that only comes from the laboratory. But the truth is, cloning actually exists in nature. For instance, many single-celled organisms, like bacteria, they reproduce asexually. They make exact copies of themselves each time they reproduce by splitting in half. In fact, in humans, identical twins are similar to clones. They share almost the exact same genes, as identical twins are created when a fertilized egg spreads in two. Now, moving on to artificial cloning, which is what we are mainly going to focus on in today's video. Animals are cloned in one of two ways. The first one is called embryo twinning. Scientists first split an embryo in half. Those two halves are then placed in the mother's uterus. Then each part of the embryo develops into a unique animal, and the two animals share the same genes. The second method is called a somatic cell nuclear transfer. Somatic cells are all the cells that makes up an organism, but that are not sperm or egg cells. Sperm and egg cells contain only one set of chromosomes, and when they join during fertilization, the mother's chromosomes merge with the father's. Somatic cells, on the other hand, already contain two full sets of chromosomes. To make a clone, scientists transfer the DNA from an animal somatic cell into an egg cell that has had its nucleus and DNA removed. The egg develops into an embryo that contains the same genes as the somatic cell donor. Then the embryo is implanted into an adult female's uterus to grow. A historical event of the scientific world was when the Roslin Institute successfully produced Dolly by cloning an adult sheep. Scientists were ecstatic because this opened a door for producing genetically modified livestock or bringing back extinct species. Nevertheless, there provided to be many negative consequences to this process, as Dolly only survived for 6.5 years, which is half the lifespan of normal sheep, before being euthanized due to a variety of complications, such as Joexiate, sheep retrovirus, arthritis, and a lung tumor. Analysis of Dolly's DNA showed that her telomeres were half the size of a regular sheep of her age, because the DNA was copied from an adult sheep, providing a potential reason for her premature death, as Dolly's DNA was double her actual age. If you're wondering what a telomere is, telomeres protect DNA from damage, and as the individual ages, the telomeres shorten and their protective properties begin to deteriorate. Now comes the great controversy. The creation of Dolly sparked a lot of debate regarding cloning and whether this should be investigated due to ethical issues and physical effects on the clone. Some people argued about the prospect of bringing back extinct species, and the scientific community saw the cloning of Dolly as a catalyst for numerous developments in medicine and biology, creating more personalized treatments for patients through the cloning of entire organs. While the advantages cloning could provide were very clear, cloning was still not encouraged and even banned in certain countries due to arguments in regards to the ethical concerns of the process if it were to be utilized on humans. Now, what is the problem with cloning? Ethics. Ethics in scientific research are of the utmost importance because science should be aimed towards discovering truthful findings and helping to improve the quality of life for humans. Following ethical guidelines in scientific investigations will help to ensure that the knowledge gained is reliable and was safely produced, adhering to basic moral and social principles. Furthermore, with ethical scientific research, the general public would be more accepting of the findings and willing to participate in scientific research, such as clinical trials. Recently, German scientists from the Ludwig Maximilians University have planned to clone and breed genetically modified pigs to use during human heart transplant surgeries. After performing this surgery on a terminally ill man, it was found that the patient responded well to receiving the heart, which demonstrates how valuable cloning could be to the human race and organ transplantation. There are many people on the organ transplant list which unfortunately do not survive the wait, and cloning animal organisms would help to reduce the number of patients on this list. However, this research received criticism from a German pressure group called Doctors Against Animal Experiments, who claimed this process was unethical, as animals should not serve as spare parts for humans. Cloning is actually neutral in terms of ethics, which can be seen from its natural occurrences, but due to the rapidly developing technology of humans and scientific discoveries, cloning has been placed in the hands of humans rather than the natural world. This means the ethics behind how and what cloning is used for now predominantly depends on us. One of the major ethical considerations as to why human cloning is not supported by a majority of the world population is because it removes the natural and human aspect of the reproduction process, while also representing a large risk of premature death. Moreover, through the use of cloning, parents would be able to cherry-pick characteristics, visual traits, or skills which they would want their children to have, which is morally troubling, as this leads to the use of decider children. Being able to choose eye color, sex, abilities, and so on with the ease of picking out accessories in a shop would lead humans to see children as possessions or projects, and would eventually result in creating children only of a certain kind. This would distort the relationship between the parent and the children. Furthermore, an advertisement for a couple seeing an egg donor appeared in the Harvard University newspaper, and it was found that they had very specific specifications for donor in terms of height, appearance, and athletic abilities, which supports the idea that humans have ideals for a perfect child, and cloning would give them the opportunity to pursue this. Additionally, it needs to be considered whether seeking human egg cells and the use of embryos is justifiable for scientific research regarding cloning. Exploiting women to obtain a large quantity of egg cells for research is a complicated process, as in addition to the few mature egg cells produced during a monthly cycle, stimulation medication must also be given to increase the number of produced egg cells. According to an article from Scientific American, there have been studies which correlate these stimulation medications with a higher risk for ovarian cancer and complications such as bleeding that can arise from the surgeries done to retrieve the eggs. Similarly to how children would be seen as commodities, paying women for their eggs would also result in the same conclusion, as this would cause the scientific community to objectify women purely as their reproductive material. During the embryo stage of early development, critical organs begin to form, which provide a sign of human life. In terms of cloning research done using embryos, this will inevitably result in the destruction of embryos, which many people view as equivalent to killing a living baby. It is our responsibility to weigh the advantages and disadvantages of cloning, acknowledging factors such as its influence on the natural order of reproduction or commodification of human beings, instead of having tunnel vision in regards to developing technology and science as rapidly as possible. The effect and principles behind human cloning can have dire consequences, and cloning, which is similar to many aspects of technology and science, is now entirely in the hands of humans and our actions. Zachary from What's Up, signing off.